Does that work okay?
Yes, we can see it.
You can. Yes, can you see it?
Yes, yes.
Okay, brilliant.
Yes, it's perfect.
That's good to know. Okay, well, thank you very much for the invitation. And I'd like to tell you today about a project that I've been doing. Actually, it's led by my colleague Bartek Václav, who's recently moved to Warsaw. And it's about modeling using computer simulations, the effects of phenotypic delay on the evolution of bacterial antibiotic resistance. So the main people who've done work on this project are a very talented ex-postdoc Martin Carvalho-Pacheco, shown here, and an ex-PhD student, also very talented, Michael Nicholson. And Ellen Lilia, who I don't have a photo of, unfortunately, she contributed a lot of biological insight to the project. So just a second. So what is phenotypic delay? Well, it's basically the time that it takes for a genotypic mutation to show its effects phenotypically. So here we have an illustration. Here's a bacterium. It's got some DNA in it, and it's sensitive to an antibiotic. And a genetic mutation happens, so its DNA changes, but it's still sensitive because there's a time delay for it to realize the effects of this genetic mutation and do whatever it needs to do to become resistant. So we're talking about this time delay between the genetic mutation and the bacterium becoming phenotypically resistant. And this is not a new concept. We didn't think of it ourselves. There's actually a very long history of discussion of this concept in evolutionary biology, and it has all kinds of different names. So it's also called delayed phenotypic expression, cytoplasmic lag, phenotypic lag, phenomic lag, and it's pretty much been discussed, well, even from before the seminal paper of Lurie and Delbruck in 1943. So it's not a new concept, but the fact that it exists for bacteria is new knowledge. So before 2018, when this important paper emerged from Sebastian Bonhoeffer's group, it wasn't known that phenotypic delay actually existed for bacteria. And what Sebastian Bonhoeffer did was to make, among other things, a very nice genetic construct where they could actually trigger a kind of mutation happening at a particular defined time and follow its phenotypic effects. And so you can see here in this genealogical tree, the genetic mutation is triggered when the red arrow happens, and you can see that it's several generations later when the genealogy goes green, and that's when the phenotypic effect of this model mutation happens. So bacteria also can show a phenotypic delay. So in this project, we did a theoretical project, and we wanted to explore what are the possible causes of such a phenotypic delay for bacteria, and also what are the consequences for the resistance, for the evolution of antibiotic resistance. And in particular, we investigated two possible mechanisms that I'm going to discuss in a moment. So one of them is effective polyploidy, and the other one is molecular dilution. And I'll explain what those are just in a moment. And we did this using computer simulations. So essentially, we did simulations of a population of evolving bacteria, including either of these two mechanisms. And we ignored the fitness cost of resistance. So we simply simulated bacteria where the fitness was equal in the absence of the antibiotic, regardless of the genetic mutation. So to discuss what these two mechanisms were that can cause phenotypic delay in bacteria that we investigated. So the first mechanism is what we call effective polyploidy. And that arises from the fact that with bacteria, they can grow at different rates. So bacteria can be fast growing or slow growing depending on the environment they find themselves in. And if they're growing fast, they have more copies of their genome than if they're growing slow. And just to illustrate why that is, so here's a schematic of a slow growing bacterium. And so as the bacterium elongates and divides, as it elongates, it copies its DNA. So it has this circular DNA chromosome, and it's gradually copying the DNA as it's growing. And then at some point it's finished copying the DNA and it separates the chromosome into two daughter chromosomes, and then it divides. Okay, and this whole process of copying the DNA and dividing the cell takes about an hour. And you can divide it into the C period, which is the time to copy the DNA, and the D period, which is the time to basically organize cell division. And so it takes an hour to copy the DNA and divide. But bacteria can grow faster than this. They can actually grow with a doubling time that's less than an hour. So how do they do that? And so it turns out that in fast growing bacteria, they actually divide before they start copying the new round of DNA. They start the new round of DNA replication before they finish the old round. So a newborn cell is born with its DNA already partially copied, as you see here. And then during its growth, it starts initiating new rounds of replication before it's finished the old one. And the effect of that is that you can have more than 2 copies of a particular part of the DNA. You can have 4 copies, or you can have 8 copies, or you can even have 16 copies. So the faster the bacterium's growing, the more copies of particular genes in its chromosome it will have. And that's actually something that's been known for a very long time. And there's a seminal theory by Cooper and Helmstetter from the late 1960s, which quantitates how many copies of the DNA you expect to have for a given growth rate, and at what time during the cell cycle the cell's going to initiate replication depending on its growth rate. So this is all well-known stuff. What's it got to do with phenotypic delay? Well, the point is that this what we call effective polyploidy, so the fact that the cell has many copies of its chromosome when it's growing fast, that can cause a phenotypic delay. So let's suppose that we have a resistance mutation that's recessive. And what that means is that all the copies of the DNA of that gene in the cell have to be resistant in order for the cell to be phenotypically resistant. So if we have a cell here that's got that's got 4 copies of this particular gene because it's growing fast, then only one of them gets mutated randomly. Then what happens is that initially the cell's sensitive because it's got 3 copies of the unmutated form of the gene. And after it undergoes one division, one of its progeny will be completely sensitive genotypically as well as phenotypically. The other one will have only half its copies of the gene mutated, so it's also phenotypically sensitive. And it's not until the second generation after the mutation that you actually get an offspring which is fully resistant because it's got all 4 copies of its gene mutated. So there's a phenotypic lag of several generations before you start to detect phenotypically resistant cells in this model. And of course, the lag will be longer the larger the polyploidy. So if the cell's growing very fast and it's got even more than 4 copies, then there'll be an even longer lag in generations. So how do we simulate that in our study? Well, we just do a simulation where we track individual cells and we track the number of gene copies in each cell. And we model the whole process of chromosome replication via this Cooper-Helmstetter theory, which tells you how many copies you're supposed to have for a given growth rate and what time the initiation of replication will happen and so on. And then in our simulations, we can vary the growth rate. And as we vary the growth rate, the number of gene copies will change via this Cooper-Helmstetter theory. And as I said before, we're going to assume recessive resistance mutation such that we only get phenotypic resistance when all the gene copies are resistant. So we're essentially simulating the process that I showed in the diagram in the previous slide for a population of cells. So here's just a kind of an illustration of how one of these simulations might work. So let's say that we have 100 bacteria which are genetically mutated at the start of our, or at least one of the copies of the gene is genetically mutated at the start of our experiment, and the population grows exponentially. Then let's plot from our simulations the probability of finding at least one phenotypically resistant bacterium in the population. So the probability that the population shows some level of phenotypic resistance. So here's some results from our simulations. So what you can see is here we've done 2 simulations with different growth rates, or in other words, different number of gene copies on average. So C equals 2 mean on average there are 2 gene copies, so that's a slow growing population. Whereas C is 4 means there on average there are 4 gene copies and it's a fast growing population. And what you can see is our simulations indeed show a phenotypic lag. So when C equals 4, so fast growing population with large effective polyploidy, we have a longer lag in seeing resistance in the population than when we have fewer copies of the gene. So our simulations do indeed do what we expect them to do regarding having a phenotypic delay caused by this polyploidy. Okay. So that was the first mechanism that we were investigating here, but it's not the only way that you can get a phenotypic delay in bacterial antibiotic resistance. So there's also another mechanism that we investigated, and that's about protein dilution. So just thinking about bacteria on a kind of protein level, essentially a bacterium can be thought of as a bag of protein molecules. And let's say that again, a resistance to an antibiotic is recessive. And what we mean by that is that all the protein molecules in the cell have to be of a resistant type in order for the bacterium to be phenotypically resistant. So in other words, what we're saying is that the antibiotic acts by making a protein molecule toxic. And even if you've only got one of them that's toxic, that's going to be enough to make the bacterium sensitive. So let's say that's the case. then if we have a bacterium which genetically mutates, so it starts making resistant proteins, initially, not all its proteins will be resistant. It's going to have a mixture of resistant and sensitive proteins. And as it grows and divides, it will gradually dilute out the sensitive protein. And eventually you start to get some progeny, which will only have copies of the resistant protein. And those are the ones which are phenotypically resistant. So the need to dilute the sensitive protein after you start making the resistant protein could be enough to cause a phenotypic delay, irrespective of what's going on with the number of gene copies. So we can also simulate this mechanism. To do that, we simply, again, track each cell, but this time, rather than tracking gene copies, we track the numbers of resistant and sensitive proteins. And we say that when the cell divides, it partitions its proteins binomially. So, there's a binomial probability of proteins going to one daughter or the other. We're going to ignore degradation of the protein molecules. We're just going to assume they're diluted. And we're going to assume that we have phenotypic resistance when all the proteins are resistant. So if we do that, then again, here are some simulations for these 100 bacteria, which are initially genetically mutated. And again, we plot the probability of finding at least one resistant cell in the population as a function of time. And what we can see is that, again, there's a delay. And the delay this time increases with the number of sensitive proteins that we start with. So if you start with 1,000, then obviously it takes you longer, more dilutions to remove them all, than if you start with a smaller number like 20. So this is not rocket science, but the key point is that in the protein dilution mechanism, the important parameter is the number of protein molecules that you have to dilute. Whereas in the effective polyploidy mechanism, the important parameter is the number of gene copies that you have to remove by division. So a question that we tried to address in this project is, considering that there are these two mechanisms for causing phenotypic delay in bacteria, do they have different consequences at the population level? So what are the effects of these two mechanisms? And we're particularly interested in antibiotic resistance. So we simulated the setup which is shown here. So an antibiotic challenge experiment. So in this experiment, we suppose that we have a population of bacteria which grow exponentially in the absence of antibiotic. And during that time, they're also mutating, as bacteria do. And then at some time after this exponential growth has proceeded for a while, we subject the population to an antibiotic such that only the phenotypically resistant bacteria survive. Okay, so that's the experiment. And just to illustrate it in a graph here, so the concentration of the antibiotic as a function of time initially is 0, and then at some critical time it suddenly increases to a high value. And then if we plot what's happening with the bacterial population, so we have an exponentially growing population of bacteria at the start, which is mostly sensitive, and it's probably got one or two, or just a few resistant bacteria. And then after the antibiotic is applied, then there's a selection pressure, of course, the sensitive bacteria die and the resistant bacteria regrow. So that's the setup. And what we did is we did many simulations of this scenario for the two different phenotypic resistance mechanisms. And we plot the probability for each scenario that the population survives the treatment. So our first result, which is kind of surprising at first glance, is that when we simulate the effective polyploidy delay mechanism, it actually has no effect on the survival probability of this population. So what's plotted here is the probability that the population survives as a function of the doubling time, so the growth rate of the cell, which of course translates into changes in the number of gene copies. So regardless of how many gene copies the bacteria have, they still have the same probability of surviving this antibiotic challenge. So it seems like effective phenotypic delay caused by effective polyploidy doesn't affect the outcome of this experiment. So why is that? Well, it turns out to be because of a cancellation of two effects, which actually, we didn't discover this. It was discovered in Bonhoeffer's study. So what happens is, as I showed before, as you increase the number of gene copies, the length of the phenotypic delay increases. But as you increase the number of gene copies, you also have more chances to get a mutation, because if you've got, say, 4 copies of the gene in the cell, then you've got 4 chances for that cell to get mutated. And it turns out that those factors exactly cancel. So due to the cancellation of these two effects, that the delay increases, but also the chance of getting a mutation increases, this effective polyploidy mechanism has no effect on the outcome of this experiment. Okay, but that's not the case for the protein dilution mechanism. So when we simulate phenotypic delay due to protein dilution, it turns out that the probability of survival of this population does decrease as the phenotypic delay increases, or in other words, as we increase the number of protein copies to dilute. So as you have a longer phenotypic delay, you're less likely to survive this antibiotic, the population is less likely to survive the antibiotic challenge. And that makes sense, of course, because as the population is growing exponentially, you have some mutants that arise late. And the mutants that arise late, when the antibiotic comes along, they haven't had time to become phenotypically resistant, and so they don't contribute to population survival. So the longer the delay, the more late arising mutants you have, which are not phenotypically resistant when the antibiotic comes along, and the smaller the survival probability. So that's kind of intuitive, I think. So why were we interested in this particular experiment, the one with the antibiotic challenge? Well, actually, one of the reasons is that it corresponds to a Lurie-Delbruck fluctuation test. So Lurie-Delbruck fluctuation test, probably I don't need to explain to this audience, but just in case. So what happens in a Lurie-Delbruck fluctuation test is indeed you let a population of bacteria grow exponentially in a test tube, and then at some time t or some number of generations, you expose them to antibiotic on a plate and you count how many of them survived. Exactly. And so that's exactly equivalent to the scenario that I just described, where we have an exponentially growing population that we subject to an antibiotic challenge. Why is this important? Well, it's important because from the Lurie-Delbruck fluctuation test, you can extract an estimate of the mutation rate. So what's done here in the Lurie-Delbruck fluctuation test is that you actually repeat this experiment many times, and you get the statistics of the number of colonies that grow on the plate, so the number of resistant mutants in the population. And of course, due to the exponential growth, you're going to get a wide-tailed distribution where occasionally you get an early mutant which has many progeny, and more often you get a late mutant that has few progeny. So that's a characteristic distribution associated with the number of colonies on this plate. And by fitting that to theoretical models, you can estimate the mutation rate. And this is how microbiologists estimate mutation rates. However, it turns out that this fluctuation test is not as simple as it seems, because it can give the wrong answer. And so this is a really nice paper from 2012 from Lee et al., where they just did a fluctuation test for E. coli. And they also measured the mutation rate by genome sequencing. So they have two independent estimates for this mutation rate, one of which is clearly correct, the genome sequencing one. And it turns out that the fluctuation test result was incorrect. In fact, it was too low by a factor of almost 10. So why is that? So probably the reason is that the model that's used to interpret the colony number statistics is probably not correct. But how is it not correct? And so we wondered whether phenotypic delay could explain that. Actually, Lee et al. Also wondered whether phenotypic delay could explain it, but what we tried to do is to ask quantitatively whether phenotypic delay could explain this result. So it's clear. So it's clear that phenotypic delay does change the distribution of colony numbers in a simulated Lurie-Delbruck experiment. So this plot here just shows some results from some simulations that we did of a Lurie-Delbruck experiment fluctuation test, where we did we simulated two different fluctuation tests, one of which had no delay, and the other one had a dilution delay with a number 16 of molecules to be diluted. And all this is supposed to be showing is that clearly the colony number distribution in these two simulations is very different. And so it's clear that a dilution delay really can change the colony number distribution in a fluctuation test. Furthermore, it actually causes a if you then try to, so you do a simulation of the fluctuation test with no phenotypic delay, then you try to do a simulation with phenotypic delay and fit the mutation rate. Indeed, you would estimate a higher mutation rate. So it goes in the right direction for explaining the Lee et al. Data. And try to do is actually directly try to simulate the Lee et al. Experiment. So from this experiment, the whole genome sequencing gave the true mutation rate, which is approximately 4 times 10 to the minus 9 per generation. And they did 40 replicates of fluctuation tests over 20 generations. So the E. coli were grown for 20 generations before the antibiotic was added. The antibiotic was nalidixic acid. And as I said, that gives a mutation rate from the fluctuation test, which is too low by a factor of 9.5. So what we did is we decided to simulate the exact same fluctuation test using the true mutation rate, which had been measured from the genome sequencing of 3.98 times 10 to the minus 9. And we then extracted an apparent mutation rate by simply fitting our simulation data to the actual experimental data from Lee et al. Okay. And we did that for varied amounts of phenotypic delay by changing the number of sensitive molecules that are supposed to be diluted in the simulation. And here's the result. So what's plotted here is the factor by which we underestimate the true mutation rate out of our simulations as a function of the amount of phenotypic delay, so the number of sensitive molecules to dilute. And so in all of our simulations, we underestimate the mutation rate because phenotypic delay, that's what phenotypic delay causes. But as the phenotypic delay increases, then we have a larger and larger underestimation of the mutation rate. And indeed, it turns out that you can make the simulation agree with the Lee et al. Data if you have 30 sensitive molecules to dilute. So the results of these simulations are that indeed, in our model, a phenotypic delay can explain the discrepancy which is seen in the Lee et al. Data between the true mutation rate and the apparent mutation rate that comes out of the Lurie-Delbruck fit. And if that's the case, then it requires about 30 sensitive molecules to be diluted in our model in order to get a result that's consistent with what Lee et al. Saw in their fluctuation tests. So that brings me to the end of the talk. So the kind of take-home message is, firstly, that phenotypic delay can be important because it can affect the outcome of models for evolutionary dynamics. And different delay mechanisms have different effects. So in the particular example that we chose, which is the sudden antibiotic challenge that corresponds to the Lurie-Delbruck experiment, the effective polyploidy mechanism doesn't make any difference, but the protein dilution mechanism does. And that possibly this might explain discrepancies that have been found in mutation rate estimates. So once again, these are the people who did the work, and especially Bartek Václav actually led the project. So thank you very much for listening, and that brings me to the end.
Thank you, Rosalind. Thank you for a great talk. We have a few questions already in the chat. First question from Joachim. How common is it that resistance mutations are recessive in this sense?
Yeah, that's a good question. So I mean, a simple answer is we don't know. And actually, Ellen, our postdoc, is busy devising an experimental system to find out whether resistance to ciprofloxacin is actually recessive or not. And it's not so simple to do that. But we think that in some cases it should be recessive. So, for example, fluoroquinolones like ciprofloxacin, how they act is they cause the DNA gyrase, which is a molecule that sits on the DNA, to become toxic by chopping up the DNA. So when it's got ciprofloxacin bound, it causes double-stranded breaks in the DNA, which don't get repaired. And so it's fairly clear that this kind of toxicity mechanism should be recessive to some extent, because if you start chopping up your DNA, even with one chopping up molecule, then that's bad, right? Whereas other antibiotics, so for example, ribosome targeting antibiotics probably don't act in that way, because what they do is effectively deplete the pool of active ribosomes, which is much more of a gradual thing. So it's possible that those ones wouldn't be recessive, but we don't have actual hard facts on that, I would say.
Thank you. Another question from Helen Alexander. Did you do any simulations combining both polyploidy and dilution mechanisms? I guess
yes,
this would be more complicated as there's ongoing production of sensitive proteins from remaining sensitive gene copies.
Exactly. So basically, you've answered your own question. So yes, we did, and I thought it was probably a bit too sort of esoteric to put in the talk. But you're quite right. So they don't just add up. There's a bit of an overlap. but it's qualitatively, you pretty much get what you would expect from combining them.
Okay, so another question. Is there a way to check whether this extra parameter, 30 sensitive molecules, is actually measured in E. coli?
Can you say the question again?
I guess the question is whether one could imagine confirming something with experimentally that the prediction about 30 sensitive molecules results reconcile with the data. If I understand the question correctly, if not,
that's kind of what our postdoc Ellen is trying to do. So she's making a construct in E. coli where you can control the expression level of the sensitive protein in the presence of the resistance protein, and then in this construct you should be able to kind of control the numbers of resistant and sensitive proteins, and then measure the sensitivity level. Another thing to say is that this model is very crude because we've just assumed that you have to have 0 of the sensitive protein in order to be resistant, which is, probably not true. So it's maybe better to think of them as kind of units than actual protein molecules. So it could be that you actually need 10, and these kind of units in our simulations should be thought of as sort of batches of 10 molecules. And of course you can do the simulation with it. You don't have to have in the simulation that 0 is the critical number. You can say that there's a threshold of whatever you want it to be for resistance. And Martine did simulations like that, and qualitatively it doesn't make a lot of difference.
Another question from Nemiko. What is the molecular mechanism for the nalidixic acid? Is there indeed a sensitive molecule?
Well, it's a fluoroquinolone. So it's one of these molecules that binds to, I think, whether it's actually DNA gyrase, it might be the other topoisomerase 4. But anyway, it binds to one of these molecules that sits on the DNA and chops up the DNA.
Mm-hmm. And a follow-up question: can't you test the model for the phage resistance, where the resistance is coming from the receptor mutation? Then clearly there is the phenotypic lag for the receptor number. Yeah,
that's a good idea. Yes, it's true that it should work for phage resistance. One of the challenges is finding a data set that's got enough replicates for well, I suppose we could do the experiments ourselves, but if we were going to use literature data, then you need to find a data set that someone's published which has enough replicates to have an accurate to be able to plot the Lurie-Delbruck distribution accurately enough to compare different models. And I'm not aware of one of those for phage, but it's true that it would be a good test system.
Great. Well, if there is one final question, type it into chat now. And otherwise, I think we should thank Rosalind again for a great talk and all the speakers in this session.